Good afternoon, and thank you for joining us for the 20th annual Celebrate Invention event at the University of Michigan. I'm Dave Repp, Associate Director of New Ventures at the Office of Technology Transfer at the University of Michigan. This afternoon, we will be presenting University of Michigan technologies and startups in the fight against COVID-19. Before we begin, I'd like to run through some housekeeping items. First, this webinar is being recorded, and as a registered participant, you will be receiving a link to the recorded event after the fact. Second, if you submitted questions during the registration process, thank you. If you didn't and have a question you would like to ask one of the panelists during the session, please make use of the Q&A feature. The Q&A feature will be monitored by our staff, and questions will be forwarded to me as time allows. I'd also like to remind you that as a registered participant, you will not be able to see the video of other participants. You will only be able to see the panelists and the video of myself. So with those items out of the way, I'd like to start off by saying a few words about the theme of today's discussion. There's plenty of debate about who said, "Never let a good crisis go to waste." And there's even some debate about the implication of that phrase. But my key takeaway is that in times of crisis, we can each rise to the occasion and use our resources to address what's in front of us. Here at the University of Michigan, we've sought to broadly disseminate research innovations that might address the COVID-19 pandemic. These include potential treatments, tests, new ventilator devices, and personal protective equipment. The university has licensed technologies royalty-free for any applications related to the pandemic. Our three panelists have certainly risen to the occasion by adapting their own innovations, resources, and know-how to solve one of the world's great challenges. So joining us today are Dr. Lola Eniola-Adefeso. Dr. Adefeso is University Diversity and Social Transformation Professor of Chemical Engineering and Biomedical Engineering at the University of Michigan. She is Associate Director of the Cellular Biotechnology Training Program, Vice Chair for Graduate Studies in Chemical Engineering, and she is co-founder and Chief Scientific Officer for University of Michigan startup company Asalyxa. Second is Dr. Sherman Fan. Dr. Fan is professor in the Department of Biomedical Engineering at the University of Michigan College of Engineering and Michigan Medicine. He is thrust leader of wireless integrated microsensing and systems and associate director of the Michigan Center for Integrative Research in Critical Care. He is also director of the NIH Microfluidics training program, and he is co-founder of University of Michigan startup company Optofluidic Bioassay. And last but not least is Dr. Yashar Niknafs. Dr. Nicknafs is a University of Michigan alumnus of the Michigan Center for Translational Pathology at Michigan Medicine, and he is co-founder and CEO of University of Michigan startup LynxDx. If the three of you would join me, that would be great. Okay, so in Monday's discussion with Kelly Sexton, one of the messages that President Schlissel emphasized to the researchers on campus is that your career is up to you and how you want to have an impact on the world. And in my mind, there hasn't been a more appropriate time in recent memory for us all to take a moment of introspection to think about our contributions to society. So before we get into great detail about your technologies and the specific of how you're using them in the fight against COVID-19, I'd love to kick things off by finding out a bit more about each one of you and the path you've chosen to impact the world. So with that, Lola, let's start with you. What was your path to becoming a professor at the University of Michigan College of Engineering, and what's driven your research interests?
Yes, thank you, and thanks for everyone who's joined us here. So I started as a chemical engineering undergrad student a few years ago, I guess, at the University of Maryland, Baltimore County, where this was around the time when biochemical and biotechnology aspect of research was coming into chemical engineering. And that was a big draw for me because of my interest in chemical sciences, but also a deep fascination for the human body and how things interact. And somehow I was able to find a happy medium in chemical engineering fluid flow principles that seems to overlap nicely with what we understand of blood flow and interactions that happen in blood. And so that's sort of what led me to some of the work that we're doing in my lab currently at the University of Michigan. So all my training were in chemical engineering, but with the eye on understanding forces and fluid flow as it related to the human body. And at Michigan, we've been working in that space, trying to understand how cells and particles move and interact in blood, which is of course what led us to the technology that we're gonna chat about later in this webinar.
That's excellent. Thank you, Lola. And Sherman, what about you? I know you spent some time in industry before joining the University of Michigan. What was your path to joining the University of Michigan College of Engineering? And tell us a little bit about your research interests. I know that you've developed quite a few technologies in your time at U of M. So if you wouldn't mind by giving us some of that background of your transition from industry to academia and kind of the breadth of your work here.
Okay, thank you, Dave, and thanks for the invitation. And so I'm a physicist by training. Actually, my major was quantum optics, which was very fancy. So after my PhD, and I went to 3M in Austin, Texas, to study photonics and also especially with telecommunications using photonics. And at that time, actually, 3M has so-called 15 free time, so you can use 15% free time to do whatever you want. So at that time, I wanted to pursue biosensing applications. So after 4 years at 3M, I came back to academia. So I started with assistant professor, and back in probably 2004, and after 15 years, I became a full professor at the University of Michigan at College of Engineering and School of Medicine. So currently, my research group has been involved in quite a few different technologies, especially we are trying to use microfluidics for biosensing applications. Microfluidics is basically we are working with any analytes in either liquid form or gas phase. And so we develop quite a few different devices for different diseases. So what I'm finding really interesting is to moving the lab research to something really useful to benefit the whole society will be much more interesting than doing the fundamental research. This is just my philosophy.
That's excellent. Thank you, Sherman.
Yeah, at 3M, actually, we say the research, we use money to generate knowledge, but, company is to use knowledge to generate money. So it's quite different.
Yeah. It's a little bit different, yeah.
Okay.
And Yashar, you've got a little bit of a different path than Lola and Sherman. I know you came out of the PhD program at the Center for Translational Pathology. So maybe talk a little bit about what brought you to the university and what you were doing at U of M, and what your course out of the university into becoming an entrepreneur has been.
Sure. Yeah. Thanks for the intro. Thanks everyone for being here. So yeah, I've had a little bit of a winding path. I came to U of M in 2011 to do an MD PhD program. So I grew up in Akron, Ohio, went to college in Cleveland, and then was real excited to be a physician. And I did a couple years of medical school. Typically with that program, you do 2 years of med school, 4-year PhD, go back and finish med school, residency, etc. So I did 2 years of med school and then started my PhD, finished that in 2017. My PhD was in cancer genomics. So mostly I was doing RNA sequencing analysis of tumors, different cancer transcriptomes, genomes, and did a lot of gene discovery, biomarker development. So it was all exciting work, but somewhere at the tail end of my PhD, I was a little disenchanted with medicine. I don't know. I think it was something about it. I think waking up really early didn't really sit well with me. It's a really stable job, medicine. So I don't know. I think I realized maybe I'm a little bit of a professional thrill seeker, and just decided to kind of jump off the cliff and start this company with my PhD mentor, Arul Cheyenne. So we started the company with the aspirations of commercializing a prostate cancer urine diagnostic test. So, and this was an extension of some work done before me, some of the work of my PhD. Basically, we had discovered that there's a couple genes that you can detect in the urine, and that can basically, diagnose prostate cancer. Can everyone still see me? Something happened on my screen.
Yeah, we can still see you.
Okay. Yeah. So anyway, so you a couple of genes in the urine, you detect them, you can diagnose prostate cancer. The assay is actually currently is available, and it, you can order. Anyone out there right now, you can order it. But when the pandemic came, we people stopped going to urologists, and that all came to a halt. So we were faced with kind of pausing for a little while, seeing where things go, waiting for, prostate cancer screening to come back up. But we were actually in a very unique and fortunate situation, because the prostate cancer test, it measures the levels of two RNA genes in the urine. And it's actually very similar to the COVID test. So COVID measures the levels of two genes in some fluid, a viral transport media that a swab has been put into, or saliva, for example. So we pretty much had the entire infrastructure in place to do the COVID test. We just needed a biosafety hood. That was like the one thing. And so we ordered one, waited for it to show up, and then just started rolling. And it was a lot of work, getting things off the ground, pivoting, running a commercial lab. There's a lot of infrastructure we had to get in place. So we started all that in mid-April. I think we ran our first clinical test at the last week of May. Since then, we've run, as of this morning, 38,000 tests. So we'll hit 40,000 at the end of this week, and that'll be an exciting milestone. So we started off in mid-April. There was about 4 people in the company. Today, we're over 35. We're approaching 40, so it was very rapid expansion. Really, we've had a great team, and it's been really, the theme of this panel is, how, rising there, adapting to COVID, or I forgot the title of it, but so I think we've been fortunate to have a team, and everyone's really passionate. It's been really cool to do work that, gets everyone fired up. I like coming to work. Everyone likes coming to work because we're able to take all of our efforts and do something that there was a clear societal need for. So we're doing a lot of testing in nursing homes. We were really passionate about serving like underserved populations. So we do nursing homes, homeless shelters. We tested homeless shelters in Ann Arbor. We did a lot of community testing. So we forged some pretty solid partnerships with the different county health departments all over the state. We did about 10 pop-up drive-throughs all over the state. And then very recently, we've inked an agreement with U of M, and we'll start doing some screening testing, a saliva test that we've developed. So that'll start in a few weeks. So it's been an exciting ride. And I guess, I'm happy to answer any questions.
Yeah, that's excellent. And congratulations on that deal. I think that's a game changer, for the university as well. So we're really pleased, that you're here, local, just down the street, and we can get test results done pretty rapidly because we've got LynxDx available to the university. So that's fantastic. We're pleased about that. And I think, a lot of what you just touched on, Yashar, is a good segue, to both Sherman and Lola. You talked a little bit about the LynxDx origin story and kind of how the prostate cancer diagnostic or assay was developed. I'd love to hear from Sherman and Lola, a little bit more about their technologies as well, because I think all three of you really started your research well in advance of the world learning of SARS-CoV-2. So none of us had that as an idea in mind. So, you know, Sherman, your company, Optofluidic Bioassay, launched a few years ago. Why don't you walk us through the develop of your novel multi-well plate technology at the university, and talk a little bit about, again, how you moved from research, you know, and translated it over to the startup into a commercial product setting.
Okay, great. Yeah, thank you, Dave. And basically, my research area is in the microfluidics. So we're trying to use microfluidics and to test all the different diseases and the markers related to these diseases. And you know, microfluidics actually consumes very small amount of samples, and also potentially can make the diagnostics very quick. And so that's why we are trying to leverage what we learned in the past in the academic, and to make something real. And back in 2015, my former postdoctor and I formed a company called Optofluidic Bioassay. And then the company is still located in Ann Arbor, probably very close to LynxDx. And so our idea is to use microfluidics and to detect the markers, proteomic markers. We are not doing gene detection. We're detecting markers using so-called ELISA. And so we developed so-called microfluidic 96 well plates, which is similar to traditional 96 well plates, but because microfluidic structure in each well, we are able to make the measurement 10 times faster than the traditional ELISA. For example, we can do the COVID-19 biomarker detection in about 15 to 20 minutes rather than 4 to 5 hours. And also prior to COVID-19, actually we've been doing the other markers detection. And so this 96 well plate actually is a sort of like the disposables for all different types of ELISA measurements. And when the COVID-19 began, and we started to run the test with N protein, S protein, and IgG on the those biomarkers in serum, and so we got some very good results, and we proved we are able to see the markers very sensitively within only 15 to 20 minutes rather than 4 to 5 hours.
That's great. And Sherman, how about kind of the launch of Optofluidic Bioassay? How did that transition occur from the technology development to really getting it into the company? What was the path for that?
Path, actually, I would say we need probably a few things. One is the right people. The people not only know the technology, but also has an entrepreneur spirit. And second, actually, we need the good market. Third, actually, is probably startup funding. And so once they are ready, and also actually our company went through NSF ICAR program. And actually, I myself went through twice, one for this company and also one for another company. Both actually are pretty good, pretty successful. So actually, this is a very good program. I was very strongly supported by the University of Michigan OTT, and to give us guidance to apply for this NSF ICAR programs back in 2013 and 2014. So that's basically, both companies were founded by my former postdoctors. They know technologies, and also they have motivations, very strong motivations to translate everything from the lab to some real applications.
That's right. Yashar can attest to that. So
yeah. Yes.
Great. And Lola, your company Asalyxa was only formed a few months ago, and your technology is quite different from the two we just talked about, in that yours is a potential new treatment for COVID-related indications. So I'd love to hear more about, again, how the technology, or how the therapeutic was discovered, and how serendipity played a role in leading to the formation of Asalyxa and what the path out was for that.
Sure. So as I said in the introduction, my lab spends a lot of time looking at drug carriers, polymeric-based drug carriers, and our interest initially was to try and understand how these particles can be designed so that they can go into the bloodstream, bind to a blood vessel wall, and deliver a drug. We just were fortunate when we started the lab, based on my postdoctoral training, that we understood how blood moves and had access at the University of Michigan to be able to do those kinds of assays using fluidics, like Sherman was describing. And so we spent a lot of time looking at how our particles move and interact with human blood cells. And in that process, we started to notice that depending on the design of our particles based on shape, size, and things like that, we can change how white blood cells were behaving in the system. And that's important because white blood cells, of course, have to interact with the blood vessel wall. They have to go out into the tissue space when there's an infection and things like that. And so that was actually the first time, at least that we were aware of, that researchers were starting to ask the question, do particles in blood alter how immune cells behave, and how can we leverage that? And we were interested in neutrophils, which are the most prevalent white blood cells in human blood. And so, of course, the first thing we wanted to do was go in an animal model to say, is what we're seeing real, or is it an artifact of an in vitro type of assay? And so we partnered with colleagues. Mike Hollister is one of our colleagues here at the University of Michigan, who has the ability to do intravital imaging. And so we were able to go into a mouse model and show that the same kinds of particle cell interactions that we see in human blood in vitro was replicated in a mouse model. And so then the next question obviously is, well, is this something potentially therapeutic? Meaning, based on my PhD background as well as postdoc work, we knew that there were situations where immune cells and their overaction can actually be negative and enhance the pathology of a disease. We see this in atherosclerosis, which is a cardiovascular disease, and we see this in acute as well as chronic inflammation. And so that the first question is there a need for being able to train how immune cells behave or redirect them? And the answer for us was yes. And so then we started to essentially pivot our lab towards that space. How can we optimize a particle-based system where we can precisely block the interaction of immune cells in inflammatory, chronic, as well as acute inflammatory disease? So that was step one. The second step, of course, is with any kind of technology that has to go into the human blood, you need something that's going to be biocompatible. So you do not want your particle to be creating sort of off-target effects, right? And so then we started to think through what kinds of materials are out there. And I had, during my PhD, been exposed to and interacted with a scientist, a colleague, and we started to talk about polymers that themselves are drugs, right? And so we landed on a technology that not only allows us to construct particles of different sizes and shape, that the particles themselves have therapeutic benefit. So it was a 1-2 impact that we ended up having. And we worked with a colleague, Stead Stadiford, in pulmonary medicine here to look at whether this will have some benefit in lung injury, acute lung injury. Because at that time, that was one of the clinical indication where there was no good therapy on the market to treat people with lung injury, and that there was a big understanding, a well mapped out understanding that the immune cells play a big negative role in that clinical disease. So we worked with them, looked at animal models, and we were stunned at how well our hypothesis and particles were performing in those animals with lung injury. And so around the time we were getting fantastic data, we were talking to tech transfer, they all got excited, maybe in my mind, maybe too excited, more excited than us in terms of, whoa. And so I'm like, okay, I guess we have something on our hands. And so we started to work with them in terms of developing a plan for further testing large animals. We were thinking of going into the pig model. And then of course, COVID hit, right? And our labs were all shutting down and we were all kind of feeling hopeless and because we can't do research. But then the story started coming out of Italy with the ventilator and how people are having issues breathing with COVID. And then you start to dig more and it started to sound like the lung injuries, very similar to what we had been working on. A lot of our focus at that time were bacteria-induced lung infection causing lung injury. And so soon enough, it came that there's a component of COVID that is immune related, that is over response of the immune system, that is leading to lung injury. And so then all of a sudden we got the aha moment. And it was around the same time that the Office of Tech Transfer was sending out their annual newsletter or something to the community of look at what we're doing at Michigan. And so it wasn't hard for people to make the connection between what we had published as a new technology and the usefulness. So we weren't planning for a company, but it just, there was a need there. And because this was an indication that does not currently have an approved therapy, so we had a lot of interest from VC companies and other groups. And so we went for it and we've been able to move this quite along because of that.
That is such a cool story. And you're right. I remember when one of our mentors in residence, Bruce Arbach, came to me and told me about what you were doing and how excited he was. So yeah, it's a huge unmet need, and you know, I think it's very cool that it's accelerated as quickly as it has. You know, from your work in the lab to where we are today. So that's fantastic. Thank you for that. So yeah, what each of you have just described are great examples of the type of translational work that's done by researchers at the University of Michigan. But what sets the three of you apart is that you and your collaborators have found a way to adapt to these especially challenging times and really leverage your know-how, your innovations and infrastructure, to make an impact and really accelerate the timeline, as we just talked about with your technology, Lola. Okay, let's set the clock back to the new year, or just after the new year of this year, and each of you was headed down probably a very specific and well-reasoned development path. But by March, it was clear to everyone that the best laid schemes of mice and men often go askew. So you really needed to figure out a way to adapt, and that's where the three of you have really shined brightly, and you've applied your technology toward COVID. So Yashar, let's go back to you for a second. Was there a specific point in time where your team decided, okay, we're gaining traction with with investors and the prostate cancer diagnostic, but we really need to pivot now? And how did you make that decision, and how did your investors react to that decision?
Yeah, sure. Thanks for the question. I think we were actually at an interesting time when the pandemic came. We had, like I said, that the prostate test is available, but it's just being run out of Michigan. So we licensed the IP and the tech, and we were in the process of moving it in-house. But because there's shared IP, we can run it out of Michigan. There's a whole process there, so there wasn't a huge rush to get it into our space. So we were actually very fortunate that when the pandemic came, we were sort of just ready to go out the gates, and hadn't just yet done it. So we actually moved into our lab space in February. So we didn't even have space at the start of the new year. So your question, like, was there a moment? I think there actually was a moment. So in February, we had moved into the space and we had started to buy instruments to get the test running in-house in our lab. Like I said, it's running in Michigan, so we were going to move it in our space. The main instrument is this qPCR machine. So you may have heard about like the COVID qPCR test, RT-qPCR. So obviously you need the machine to do that. So we had a lot of other things in place. We had ordered this qPCR machine, and it was supposed to arrive in like 8 weeks. So when the pandemic struck, we were all like, okay, I was talking to Arul, and we're like, well, it'd be nice to help out, but it's going to take 8 weeks to get this qPCR machine. So, let's just sit back and kind of enjoy the ride and see what happens. And then I don't know what happened, but the qPCR machine just showed up like 3 days later. And I think we had gotten a certain model that is like a different type of a machine. And then everyone started, I think, probably canceling orders to get this specific like FDA approved machine. So our machine just showed up. And then we were kind of faced with this, I called rules, like, hey, this qPCR machine showed up, and I was kind of enjoying the quarantine, just kind of sitting back. And that was, I think there was a moment where we're like, all right, well, we just got to do it. And so, we had just closed our initial fundraising round in January. So we had a little bit of money in the bank account. We sat down, looked at the logistics, how much capital is this going to cost? In all candor, we got, because you got to put in, an investment to get this thing built up, to get off the ground. So, from a cash flow perspective, we were treading water for a little bit, and it was a risk. And so from the investor standpoint, we had reached out to a couple of people that were kind of interested and we had been circling around with. And I think it was an interesting time because no one really knew what was going to happen with COVID. And I think a lot of people were, protective of their pocketbook at the time. So we were hoping to get, maybe a little bit of, a bump, some bridge money so that we could get the COVID testing off the ground. And people didn't know really how to deal with that. So we just kind of took a risk, decided to do it. And we were really just operating under a, if you build it, they will come mindset, knowing that there was this, day after day headline news article about there not being enough testing. Just really just trusting that we could find it. And, interestingly, when we started, we were live, like I said, at the end of the May, we ran our first tests, and we were running like 20 tests a week, for like 2-3 weeks, 20 to 50 tests. We just could not get tests. It was really hard to like get into the right places, get connected with the right people. So there was a little bit of a time where it was a little dicey, but things took off.
I love that story, and I love the fact that again serendipity played a role in that the PCR machine just showed up, and maybe you're right, maybe it was sort of fortune shining on you because other companies, other labs were ordering different platforms, but that's a great story, and I love, again, I love how it just worked perfectly from a timing perspective. So and Lola, going back to you for a second, so you already had mouse data for acute lung injury kind of in that time frame that really looked quite positive. So the link to COVID was pretty straightforward, one might say, but what is it about your technology that really makes the leap to COVID and maybe getting into a clinical study in COVID, kind of the right path at this time, or the right first indication at this time? What's the synergy?
Yeah, that's a great question. I think I alluded to that earlier, in that because we spent time to think about our technology from the framework of potential rapid clinical use, we wanted to use or to construct our particle technology from materials that can quickly go into humans, meaning they have a pretty good safety profile. Because with particle-based technology, one of the biggest issue that you have is you can make all kinds of fancy stuff, but when it comes to it being a polymer system that is a high molecular weight, there's always concern in terms of how is this going to break down in the body, what are the toxicity issues and things like that. We just happen to think through that earlier or sooner than later. So in the development process, we decided to go with a technology that allows us to actually take a simple drug that is normally used that for decades now in human, and we synthesize it together to make it into a polymer, which then we constructed our particles from that. So essentially we have a solid particles in the nano range and submicron range that can be injected, that can serve to interact with immune cells. But when it breaks down into drugs and material that is already being in humans. And so part of the excitement and part of how we gain quick traction was because we had something that has what we would say a very good safety profile in terms of how it might behave. We know how every component of our technology works in human.
And how are you leveraging the COVID pandemic to kind of accelerate the process of getting into the clinic? You know, what's the key regulatory issue that you're able to take advantage of so that you can potentially get this into humans sooner rather than later?
Yeah, I think the biggest opening for us was the fact that a lot of how patients were being treated with COVID required, at least at the end game, was ventilator. And that was the biggest issue in March, April, May, where as the number of cases were going up, you start hearing hospital capacity full, ICU capacity full. And what that really means is we ran out of ventilator and we have to kind of wait for somebody to die or to recover to get somebody else in. And so because our technology was positioned in a way to mitigate the lung injury that ends up leading people into ventilator, it was an angle that we can go to regulatory FDA and folks like that to have them look at our technology. And because we were also going after the piece of COVID, acute lung injury, acute respiratory distress, that is a clinical disease that occurs regardless of COVID, right? All kinds of infection, bacterial lung infection, pneumonia, sepsis, all can cause this disease. And there really, again, isn't anything out there that's good enough to have an impact clinically in this disease. And so we leverage sort of this idea of that there is nothing out there, as well as in this health crisis, we have something that could help alleviate the shortage that could end up happening if we get an increase in rates and an increase in need for hospitalization. This is a way to sort of ease that burden on ICU. And so from there, we were able to get into, we were able to get in with the regulatory folks to have conversations about our technology and getting guidance early on because of that.
That's fantastic. And Sherman, was optofluidic bioassays shift more evolutionary, or what is, was there more complexity to applying your technology to COVID assays than just merely a shift in reagents? What does the kind of shift in strategy look like for optofluidic bioassay?
Okay, I see. Actually, before COVID-19, we have a product in the beginning of mass production. At that time, actually, we have we were in a negotiation with a large company, bio company or life sciences company, for the RUO market, research use only market. So for this COVID-19 actually basically accelerated our process to go into the clinical market. Typically for this, like, the devices, typically people go with RUO market first, then after a couple of years of try, and then they will go probably to clinical market. But this COVID-19 pandemic basically helped us to accelerate the process of clinical validation. So basically, we've been doing this biomarker detection for quite a long time. So I would say our device actually is sort of like one size fits all. So it can detect any different all different types of markers, and COVID-19 just one of them. So we just changed the reagents, and of course, the detection of antigen, like M protein or S protein shedded by COVID-19 virus, is different from detection of antibody, which like IgG. So it's just to change the protocols. Of course, we spend some time figure out the different protocols for different analytes, and then then after that, then everything is quite straightforward. And so basically for us, actually, it's not a big pivot. It's basically it just changes the protocol and also accelerate the process from RUO market, shift the resources a little bit from RUO focused market to clinical, especially COVID-19 focused market.
Gotcha. So, before we shift to some of the audience questions, I'd like to ask each one of you what this pivot means to each one of your companies, maybe for the longer term. We all long for the day when we can get back to normal or whatever the new normal is. But as you think ahead to your business strategy, 1 or 2 years from now, how has this shift affected your business plans? Do you think you'll go back to the same plan you had in February or pre-COVID, or does it require a radical rethink? And Sherman, let's stick with you because you talked a little bit about some of the partnering discussions that you have ongoing. Can you talk more about those in the longer term plan?
In the longer term, probably we will stay as the same, because we're going to move into clinical market anyway. So it's just the COVID-19 just makes things happen just faster and sooner. So we will probably stay more or less the same strategy, and just probably make something happen earlier and sooner. For example, like clinical test or clinical validation.
And Sherman, I know you're also partnering, with some other entities, and you've got some technologies that maybe are still in development, like technology that does breath analysis. Will that kind of layer into your longer term strategy, and what is that technology?
Breath analysis actually is a different technology. And also, it's going to be a different company. Actually, we formed a company just maybe a few months ago, and at that time, actually, the breath analysis was for cancer and also ARDS detection. As Lola mentioned, acute respiratory distress syndrome, and so we hook our breath analyzer to the ventilator and to monitor the patient's breath non-invasively every hour up to 10 days right now. And also COVID-19, of course, for breath analysis, actually, for all types of breath analysis, actually they are the same. So we get all markers, and then it really depends on which disease you are targeting. We have a different algorithm on software level, but not on the hardware level. Of course, COVID-19 gave us a pretty good leverage for this breath analysis to go to the market earlier than what we had planned before.
And Yashar, what about you? I mean, you're doing something completely different, at least on a day-to-day basis, than maybe you thought you'd be doing, at least in terms of what your assay is detecting. But how does this affect your long-term plans and what your investors had in mind for the company?
Yeah, this is a great question. I think it's been really interesting. This has been a really interesting experience for us. And I think it's actually probably had a pretty dramatic shift in our long-term business strategy. When this first started, we were a small operation. We had a handful of people, and we had a test that was already at market. It was already getting reimbursed. And we really just had to kind of get people believing in it, get people ordering it. So our main focus was on, getting evidence for the test, lobbying for insurance reimbursement, and then sort of starting a small grassroots effort to get people ordering it. All the urologists that we knew, there's a lot of people that were interested, just people hadn't heard about the test. So we just really needed to kind of market it. And then, obviously expand beyond a grassroots effort. But really the initial goal was, hey, can we get enough people ordering this test to get enough traction to show that it's legitimate and there's a need for it and a demand for it. And at that point, ideally get acquired by a larger diagnostics company that could really take this thing to the moon. Because like I said, we were a mom and pop shop, handful of people, mostly, in concept. We didn't even have the quant studio. We didn't have the qPCR machine. So it was really just kind of a smaller focus of, okay, let's let the big boys come in and take this thing off the ground, and let's just kind of tee it up. Today, we're in a much different position. Today, in less than 4 months, we ran 40,000 clinical tests. And the thought of running one clinical test in March was a daunting thought. I was like, man, just talking to the team and we're gonna have to get billing in place, just reporting, CLIA validation, all this stuff. And it just seemed like such a bear. And so the thought of doing super high volume testing, because if the prostate test takes off, we'd want to be doing tens of thousands of tests a year. And that just seemed like a really daunting task. Today, it doesn't. We've been able to grow. We have the people, we have the experience. We've really proven ourselves and shown that operationally we could do this. So I think that's been a pretty big shift. We've had, barely have time to catch our breath here. So there hasn't been a lot of extensive discussions on long-term strategy, in all candor. But when it does come up, I think there is an excitement because we now, it's a different vibe. It's a different feeling of like, what? I don't know why would we have to give this test out to a bigger diagnostic company? We can do it ourselves. And that was, we've been empowered by the experience here. And I think that, for the better, I think that this will alter the trajectory of what we're planning on doing in a great way. We're no longer dependent on or need someone else to come in, parachute in and help us blow this thing up. We've blown up a COVID test. So, all right, different test, different market, but let's do it. So that's been a big shift. And I think from our investor standpoint, I think everyone's pretty happy because, we went from, small operation, hoping to make some revenue someday to, revenue fueled, cash flow positive, ready to take on the world. So it's been great. I've been very grateful and every day is exciting.
It's fantastic. And Lola, obviously your company Asalyxa didn't exist pre-COVID. So I'm not sure there's a pre-COVID strategy to the company per se, but certainly you had, a different strategy maybe ultimately for your technology or, your research longer term. How does this whole event space affect your thinking long term personally, as to how you want to commercialize your technologies and then this specific technology that's been licensed to Asalyxa?
Yeah, so I think you're correct. We didn't anticipate COVID and the acceleration it gave us. But I think the idea of the technology and directions we want to go with it, interestingly, has not changed, because unlike the other two colleagues that I have here, we didn't really have to do a shift because we were focusing on a disease that is upstream of many clinical indications. So in the case of COVID, it's the ventilation that has to happen that we're trying to get in the way of. And so we had already a list of diseases that we were going to go after that were inflammatory in nature, and trying to apply our anti-inflammatory therapy to. And what makes us unique in this game is that we're not going after a singular cytokine or a singular marker. We're going after the cells directly. And so then any area where we can target the cell and get them out of the equation, we can play. So what the COVID environment has done actually is highlight to the world this big clinical problem of ARDS. It's existed for several years. The clinicians that treat patients know it's a big problem. The general masses did not know. So unless you were in the clinic having to be put on a ventilator, or your family, most people don't understand ARDS or that this was a problem. And so this sort of global light shining on ARDS allows us to be in a better position for fundraising, because now you have investors who see that this is a big deal. And not only that, for better or worse, there will be other novel viruses that will come down the pike. And if they are respiratory in nature, it's possible, we hope not, that we could be in this space again a decade from now, where it's a novel virus that got out of our hands, and then it's creating lung injury and things like that. So the profile of SARS-2 is not, I mean, it's very reminiscent of SARS-1, except that we were able to bottle up SARS-1 before it got out of hand. And so from that context, we are still, our technology is here to stay in this, to the extent that we have a successful clinical trial, we can still have an impact in this viral pandemic as well as future ones. And then of course, again, this is a clinical need anyways before we got COVID-19. So our game only changed because COVID-19 highlighted to the world, this is a big clinical need, which we of course have leveraged in our fundraising and has propelled us to this point.
Excellent. So I do have a question from the audience, and so it's a bit of an expansive question, and I'll throw it out to all of you. But really the question is, given how decentralized our response to, how decentralized academic research is in the United States, it may not really lend itself well to a coordinated response to a pandemic like this. So given your experience, do you think there's a way to do it better the next time around? Lola, you just described, the fact that there probably will be a next time, and the time horizon may not be too far away. So is there a better way to coordinate a public health issue like this so that it doesn't rely so much on some of the things like serendipity that we talked about before, or is the system working as it should?
So I actually think that the academic space in terms of technology is not so decentralized. I think the Office of Technology Transfer do a pretty good job, I think, of keeping track of technologies that we have at U of M and their application. And so early on, some of the investors we were talking to with our technology were talking to colleagues of mine that have antiviral, for example, and all of those conversations was facilitated by the Office of Tech Transfer, because they have an eye on all the technology that's coming out of U of M. So I, as an individual, may not know the next guy and what they're doing, but Office of Tech Transfer is connecting us. So I remember conversations where we're talking about our technology and they're saying, well, you know, so-and-so in pathology has something similar. I wonder if you guys could talk. So they are connecting us in ways that I think if we focus on what we've learned during the pandemic, and we have this Office of Tech Transfer working, even putting more energy in those kinds of connection and conversation, we're actually already set up that we can have a centralized response. At least that's my view.
Thank you for the plug, Lola. I appreciate it. The check is in the mail, but no, I think you're right. You know, I do think it's, you know, it's the responsibility of tech transfer offices not just to coordinate, you know, inside their universities, but also to facilitate connections to other university tech transfer offices, investors, and industry. So it's a really good point. I appreciate that. So we're almost out of time here. So, you know, maybe, you know, going kind of around the table one last time, spending 30 seconds with each of you. Do you have advice for researchers at the university who are interested in forming a startup based on a technology that they've developed in their lab? You know, what one key piece of advice would you have to those researchers? And Sherman, you've been in this game a long time. Maybe you can field this question first. You're on mute, Sherman.
I have some experience with different companies, large companies, 3 or 4 different small companies. And so what I'm thinking is actually the important message to the researchers in academia, in academic institution, is find the right people who is not only doing the good research, but also should have a good entrepreneurship spirit, and also strong motivation. So that's something really, really important. I mean, publishing the paper, I mean, to me, actually, it's probably easier, but actually doing the business actually is much more difficult. It's a completely different scenario. And so I would say, yeah, generating data in lab doesn't mean you have a good business. So finding the right people and form a very good team and work really hard, that's something really, really important. Actually, for example, one of our technologies were waited for like quite a few years before we licensed to another company, and in the 3 years they commercialized it already. So basically, they can actually. I couldn't find good people, but they found it. They found a good team and good investors, and they started. I waited for this, you know, like a good combination for quite a few years, and they stopped by my lab and they stayed in my lab for 2 days, and after 3 days after they left, they wanted to start up a company, and they licensed technologies.
Having a great team always helps.
Yeah, that's something really, really important.
Yeah. Good advice. Lola, you know, 30 seconds from you. What's your key piece of advice?
I think what made a difference for us is the great partnership we had with the mentors in residence with Tech Transfer, like Bruce. So having an expert who's gone through several iterations of taking a drug from lab to clinic guide us early on. And honestly, for us not to get all mentally wrapped up, our ego wrapped up in, you know, being able to listen to their advice in terms of, you know, if you do this and do this type of animal work, this will help us really greatly. And us being open to those feedback, because as Sherman mentioned, we're only used to writing papers and scientific process is different, but for commercial space, what they're looking for, the milestone they're looking for is very different. And so having some sort of expert who's gone through the process on the commercial side early on becomes very beneficial.
And Yashar, what about you?
Sure, yeah. A lot of the, drawing from what Lola indicated, you know, some similar concepts. And when I started this, I knew nothing about business, absolutely nothing. You know, just got my PhD and was ready to go. I think that things that were invaluable along the way that have enabled, you know, me and the company to get here, you know, building on what Lola had indicated, is just leaning on good resources. And if you are, you know, anyone out there listening, if you're with U of M, we have the luxury of a robust, willing to work, well-resourced tech transfer office, the mentor in residence program. You know, we had, you know, Jim Arthur's, shout out to you if you're out there. He was our mentor in residence, just an incredible help. So, you know, like early on, I think, you know, lean on good resources and then find good mentorship early on. And then I think, you know, coming from science, I think a big part of it was humility. You know, because a lot of, you know, if you got a great discovery and you're starting a business, you're probably successful in science. And when you're, you know, science has got its own world, its own ecosystem and a culture. And when you're successful in science, you know, you get clout, you get cred. You know, I could go give a talk. I was confident in it, and people could ask me questions, and I could respond to them effectively because I knew my work and I did good work, and I was a big fish. You know, and I was very confident in that. And if you're good at science, you're probably smart as well. I think there's a danger in potentially overestimating business acumen. You know, there's this feeling like, oh, I can do anything, I'm smart enough. And I think that having a humility and a willingness to accept that this is an area of expertise where you're probably going to have to lean on someone else to help you get through it. And I was very fortunate to have, you know, a few individuals, or my COO he's just my guy, incredible asset. And it was really just like, you know, feeling comfortable being a small fish now, feeling comfortable not knowing anything. You know, I didn't know what a convertible note was. Yeah. You know, I didn't know anything, but now allowing myself to be comfortable with that to the point where I could learn rapidly and grow, I think was really a key piece.
Yeah, let me add one more piece of advice or
Okay.
maybe suggestion. One is actually OTT is really good. I would say especially at the early stage and for newcomers, they are very, very good and to connecting us to the external world and also connecting us to some other people. I mean, we usually do not encounter or even interact. Okay. Second, actually NSF iCore. And so this is something actually, especially for you know, someone who is interested in doing something like starting up a new company. I mean, I went through NSF core program twice. It's eye-opening for me because I look at the things differently. Initially, I thought everything should be very perfect to sell a product, but that's not the case. Everything to be perfect to publish a paper, but it cannot sell a product. You know, so it's a very, very good training for me. Actually, I still remember like 5 years ago, you know, I was, you know, it's completely mind-blowing. So I recommended OTT, really interact with OTT intensively, and also if you have opportunity, try you know, one of NSF iCore programs.
See, I should have asked this question earlier. I've gotten the three of you all fired up. So, but unfortunately, we're 3 minutes over. And I'd really like to thank the three of you for contributing your time during what's an especially frenzied time on top of a usually busy time of year for you. It really means a lot to have the three of you here. And I'd also like to thank the registered participants who have watched this session and submitted questions. Please enjoy the rest of the afternoon and the rest of the sessions this week, and go blue.
Okay, go blue. Yeah, thank you.
Goodbye.
Bye-bye.